Eat general cells with because I'm going to
Friday.
Hallelujah.
Should have a quiz today. So those of you who went to the public health thing, learn anything? I don't hear any overwhelming response. Okay. Quiz Monday, 7173, which is information that we will definitely have covered already. Nothing? Okay. So, what's the correct answer? I see one correct answer. I see another one. Another one. Several of them. Okay, correct answer is number 1. Fermentation does recycle reduced NAD back into oxidized NAD. Is it the same as glycolysis? It contains glycolysis. Glycolysis is a part of fermentation, but remember that there is that extra step or steps that have to be added on in order to accomplish this. Does it only occur in yeast? There's a lot of cells that ferment, including bacteria, muscle cells. Does it produce more ATP than glycolysis? How many does fermentation produce?
4.
How many net does it produce?
2.
2. How about glycolysis? How many does it produce?
2.
2. Okay, so no net gain in ATP produced. Okay, this is really a fundamentally important metabolic pathway. We cannot have aerobic cellular respiration if we don't have glycolysis occurring first. Because aerobic cellular glycolysis is what generates the pyruvate, and then pyruvate will enter the mitochondria if there's oxygen present, and then be converted into acetyl-CoA, and then we're going to go into the citric acid cycle, which we'll talk about today. Which pathway is more ancient, evolutionarily speaking, do you think? Glycolysis or citric acid cycle? Which one came first?
Citric acid cycle.
You got some difference. Why would you say citric acid cycle?
I was just thinking since mitochondria came from different organisms, they're periodic, and then they enter eukaryotic cell.
Okay. Why do you say glycolysis?
Just think the opposite. It's like we didn't always have organelles like that, or like not everything has organelles like that.
Okay, yeah, the first cells were prokaryotic in nature, didn't have organelles. Citric acid cycle, we'll see in a minute, is not going to happen if there's no oxygen. Was there always oxygen in our atmosphere? No, that was a more recent phenomena than the origin of life. It wasn't until we had photosynthesis evolved that we had oxygen produced. So we think that this is really the most ancient metabolic pathway, and that these metabolic pathways evolved later in response in part to things like accumulation of oxygen in the environment, which then gave us a more efficient way to process glucose and get energy. Remember that if we do all of this together in aerobic, we get how many ATP? 38 max, okay? And glycolysis would only generate the 2 net, okay? I know that all this stuff seems kind of esoteric sometimes, why am I having to mess with this and learn all this stuff? But just kind of think about it for a second. These processes are why you're alive. Okay? Every cell in your body has to be doing this 24/7, 365. And if the process is stopped for even a few minutes, your cells start dying. And so these metabolic pathways are literally the difference between life and death, cell organization and cell disorganization. Contemplate your arm or your hand for a minute. Think about how many cells there are in there. And every one of those cells, these chemical processes are happening. Anybody have any idea of what the estimate is of the total number of cells in the human body? Even just a wild guess?
How many cells?
Cells.
Like a billion?
Way more.
No.
100 trillion.
Well, yeah, no, I mean, I've seen some estimates as high as that. I was just looking before I came in here. And it seems a lot of the estimates are hovering around 50 trillion human cells.
There's a lot more bacteria.
But, yes, but the human organism consists of about, if you believe that number, 50 trillion human cells and maybe as many as 100 trillion bacterial cells. You are only about 1/3 you, if you look at your whole organism. You're about 2/3 bacteria and about 1/3 you. And those bacterial cells are all doing this too. They can all ferment, and many of them can carry out this aerobic cellular respiration as well. So this is a very significant process, and it's very relevant to your existence. Okay? Okay, so pyruvate generated from glycolysis enters the mitochondria and is preparing to enter this sequence of reactions called the citric acid cycle. So let's take a look at first, kind of in general, what's accomplished in the citric acid cycle, and then we'll come back and talk about it in a little bit more detail, give you some names of some compounds, but again, don't get all hung up on the names because those aren't things you're responsible for having memorized for the exam. Okay? One of the things we like to do when we talk about metabolic pathways involved in respiration is count carbons. Okay? So pyruvate is a 3-carbon compound. Now remember, a glucose gives you 2 pyruvates as a result of glycolysis. Okay? So when we start kind of adding everything up, and we start seeing a reduced NAD here, one here, ATP produced down here, that's per pyruvate. So to account on things on the basis of a glucose, you really have to double those numbers. Make sense? Okay. So we talked about this a little bit last time, this reaction that prepares pyruvate by converting it into this molecule acetyl-CoA. And this is the entry point as far as the citric acid cycle is concerned. So remember the list of things I asked you to be familiar with and understand for the exam. What's the starting compound of the citric acid cycle? We would consider acetyl-CoA to be the starting compound. Okay? This complex, the pyruvate dehydrogenase enzyme complex, is doing a lot of things to that pyruvate. It is decarboxylating it. It's removing carbon in the form of CO2. Okay? So when you exhale CO2, that's where some of that CO2 is coming from, is this reaction. Remember what we call this reaction? We call it a transition reaction. Okay?
Does that happen inside the matrix too?
Yep. Happens in the matrix of the mitochondria. So there are enzymes. Pyruvate dehydrogenase is sitting in this matrix of the mitochondria. Okay? It's dehydrogenated, so some hydrogen stripped off of it. So we get some reduction of NAD. And then this coenzyme A is temporarily added onto it. We're going to see later down here, when we look at this in more detail, the coenzyme A is later taken off again. So that coenzyme is recycled and used over and over again. So we go from 3 carbons to 2 carbons. Now, remember the equation for aerobic cellular respiration. What were the 2 substrates? What were the 2 starting materials? Glucose and oxygen, right? What are the end products? Carbon dioxide, water, and some energy. Okay? So we're going to have to convert all of the carbon that's in glucose, how many? 6. Into CO2 molecules. So there should be 6 CO2 molecules produced. Okay, when we add everything up. So here's one of them per pyruvate, or 2 per glucose, right? Here's where the remaining 2 come off of pyruvate. So 2 per pyruvate, 2 more per glucose. So 3 times 2, that gives us our 6 carbons that were originally in glucose. We completely oxidize the glucose and remove the hydrogens, and we're left with CO2. One of the first reactions involves removing this CoA from acetyl-CoA. And when I show you the names of compounds, there's got to be something in the citric acid cycle that grabs the acetyl-CoA and pulls it into that set of reactions. At some point in time, carbon dioxide is evolved as we oxidize the acetyl-CoA. Where do those hydrogens go that we remove from the acetyl-CoA? They go to some NAD. And then there's another coenzyme involved only in the citric acid cycle called FAD that can be reduced as well. The enzyme that works over here requires this coenzyme. There are other enzymes that require this one. Okay? But NAD and FAD behave the same way. They pick up electrons and become reduced, donate them and become oxidized. Sorry. We do produce a little bit of ATP down here. What process produces this ATP? We got two ways you can make ATP. What are they? Substrate level phosphorylation and oxidative phosphorylation. This one's happening just in the matrix of the mitochondria. It's not associated with any membranes. So what is it? Substrate level or oxidative? It is substrate level phosphorylation. Okay? One of the things about cycles, like the citric acid cycle, is that if this is going to keep working, the thing you start out with always has to be regenerated. So during this process, whatever molecule picks up acetyl-CoA, when we come back around again, that molecule has got to be reproduced by the cycle. Okay? So if this keeps regenerating itself, we keep inputting carbons 2 at a time into the cycle from pyruvate. And we lose those carbons during the cycle in the form of CO2. Okay? So we keep funneling in pyruvate from the breakdown of glucose during glycolysis. Convert that pyruvate into acetyl-CoA, because that's the entry point into the cycle. And then we remove those 2 carbons in acetyl-CoA, and things keep turning around. Everything.
So glycolysis makes 2 ATP, and a citric acid cycle makes 1.
1 per pyruvate, or 2 per glucose.
Okay.
Okay. So now, boy, we've doubled our ATP now, right?
Okay.
We've gone from a net of 2 to a net of 4. Because are we using any up in this sequence? Nope. Nope. Only during glycolysis do we use the ATP initially. Okay, so here's our pyruvate from glycolysis out of the cytoplasm. Here is the membrane of the mitochondria. So we've got a transporter protein that is going to transport that pyruvate into the mitochondria. And it's eventually going to be transported into the matrix. Okay. Pyruvate is converted into acetyl-CoA. Okay, so we get a CO2 removed. We add some coenzyme A. We reduce an NADH. And remember, these are outcomes per pyruvate. So then we have acetyl-CoA. And this becomes the entry point then into the citric acid cycle. Okay, again, now, don't get hung up on structures or names, except possibly this one. Okay? I told you acetyl-CoA is kind of the starting point. But I guess we also have to consider this compound also a starting point, because that's what grabs the acetyl-CoA and then feeds it into the citric acid cycle. So this compound is called oxaloacetate. So this and this are kind of the starting points of the citric acid cycle. Let's count carbons. 1, 2, 3, 4 in oxaloacetate. 2 in acetyl-CoA. When those 2 are stuck together, a 6-carbon compound is produced called citrate or citric acid. That's where the name the citric acid cycle comes from. Okay? So what's going to be accomplished in the whole rest of this cycle? Because remember, we always have to get back to here. We always have to get back to oxaloacetate. So that can pick up more acetyl-CoA and it can go again. Well, what's going to happen is 6 carbons are going to be converted into 4 carbons. So we're going to lose 2 CO2s. We're going to remove some of these hydrogens, and so we're going to reduce NAD and reduce FAD. And then we're also going to make a little bit of ATP by substrate level phosphorylation. Okay. So, oxaloacetate 4 carbons grabs acetyl-CoA 2 carbons and form the 6-carbon citric acid or citrate compound. And then there's an enzyme that basically converts citrate into an isomer called isocitrate. Don't worry about that. It's just getting it ready to do another reaction. Isocitrate, which still has 6 carbons, is decarboxylated to form this compound called alpha-ketoglutarate. Not only is it decarboxylated, but it's also dehydrogenated. Okay, so some hydrogen is removed here. And that hydrogen is used to reduce NAD. Alpha-ketoglutarate is converted into succinyl-CoA. We've got this weird coenzyme A here again. Okay, so we have some more coenzyme A being stuck onto this molecule. Also, another decarboxylation is occurring. So this has 5 carbons, this has 4, so we're losing another CO2. We've now lost the 2 carbons that we fed in here and here. So we're back to a 4-carbon compound. This reaction is also an oxidation, so we reduce NAD. Well, what's the rest of the steps over here involved in? Basically getting this compound back to this one again. And along the way, we get some additional energy payoff. So, succinyl-CoA is converted into succinate. 4 carbons, 4 carbons, okay? We lose that coenzyme A again, okay? And then a substrate level phosphorylation occurs. This is, when this is converted into this, a phosphate group, which is actually an inorganic phosphate, is grabbed and stuck onto GDP to make GTP. And we haven't talked about GTP yet. GTP is kind of a cousin of ATP. Remember what ATP stands for? Adenosine triphosphate. Okay? It's very similar to that adenine nucleotide in DNA. What does GTP stand for? Guanosine triphosphate. Very similar to the G nucleotide. Cell's kind of interesting. For some cellular processes, the cell uses ATP as an energy source. For some cellular processes, like transcription of, or translation of proteins, it actually uses GTP. The product that's produced technically is GTP, but then that immediately transfers its phosphate to ADP to make ATP. That's kind of a picky detail. And as far as I'm concerned, as long as you know that ATP is finally produced by substrate level phosphorylation in the citric acid cycle, this is just kind of a curiosity. Don't get hung up on that detail. Okay, succinate, a 4-carbon compound, is oxidized into fumarate. Some of the hydrogens are removed. This enzyme right here requires the coenzyme FAD as opposed to the coenzyme NAD. So we produce some reduced FAD. Fumarate is converted into malate. We have to add some water molecules to it. Still 4 carbons though. And then malate is finally oxidized back into oxaloacetate. So we produce some additional reduced NAD. And then oxaloacetate can pick up 2 more carbons from acetyl-CoA, and we do the whole thing again. And every time this cycle turns, for every one of these we feed in, these are the outputs. Okay? And remember, this came from a pyruvate. So if we're adding everything up in an accounting ledger of energy, we've got to double everything on this slide to account for a glucose. Okay. Well, I kind of facetiously said, oh boy, we doubled our ATP yield from 2 to 4. But what do these represent right here? They represent potential energy. And if we can recycle them back to their oxidized state again, not using fermentation, but instead an electron transport chain, now we can make some ATP energy out of those.
What did you say it was that recycled? That was possible ATP?
Okay, if we've got a bunch of these, right? Accumulating.
What is it? The NAD?
Reduced, 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 reduced. Okay, we're accumulating a bunch of those. At the expense of these. Okay, so we're running out of this pretty fast. So we do need a recycling mechanism. Well, could we run them through some kind of fermentation process? Well, I suppose theoretically you could. But remember, that last step in fermentation just recycles without producing any ATP. So that's kind of a waste. But if we recycle these using this electron transport chain, which we haven't talked about in detail yet, then we can get some real payoff of energy. Do you remember how many ATPs I said each one of these represented if you ran them through the electron transport chain? 3. This one's a little bit different. If you run this one through the electron transport chain, you only get 2 ATP. That's something you're going to have to remember. Okay? So this is 3 potential ATP. This is 2. So if we take the transition reaction along the citric acid cycle, look at the potential energy we could produce from electron transport. Okay? We make 1 of these per pyruvate, or 2 per glucose. So how many potential ATP is that? 3 times 2, remember? Okay, this is per pyruvate. There's 2 pyruvates. So that represents 6. How many does that represent? 18, right? 3 times 3 times 2. How many does that represent? 4, okay. 2 of these per glucose. 2 ATP per each one of those. So you start adding all those up, and that's really where you get your energy payoff. Plus, if you've got those 2 reduced NAD from glycolysis, and you don't ferment them if you're doing aerobic cellular respiration, they can be fed into the electron transport chain as well. So that's how we jump from 4 ATP by substrate level phosphorylation to 38 total, is by generating all these reduced NADs and FADs and then feeding them back into the electron transport chain to recycle them, and in the process making that hydrogen ion gradient, charging up our battery if you like, and then using that hydrogen ion gradient energy to make ATP by chemiosmosis. That's where we get the payoff. So again, don't get confused and overwhelmed by all of these names, and particularly the structures. If you were taking an upper level biochemistry course, then you certainly would probably have to know the names of everything in the citric acid cycle. If somebody was really picky, you might have to know the structures of all of those too. I've never been a big person on memorizing structures, but I think it's important to see them and to kind of look at them and see what's happening in metabolism. I think part of my aversion to memorizing structures was in grad school. I took a course called Intermediary Metabolism. And every exam was just a set of metabolic pathways. And you had to write, okay, glucose, and then you had to draw everything out, including all the structures, all the enzymes, all the coenzymes and everything. And I would just fill up boards memorizing, and as soon as I walked out of that class, I dumped it all. Okay? I don't really want you to do that. So I think that looking at these and kind of seeing what the arrangements that are occurring or happening is fine. But I want you to understand more big picture here now than all of this detail, which just makes you forget the big picture. Okay, so now we're ready to talk about electron transport. Where does the electron transport chain exist? In this inner membrane of the mitochondria. Okay, so we've got this membrane, all these chairs around the perimeter, surrounding the matrix. I'm standing in the intermembrane space, and then out here the walls are the outer membrane of the mitochondria. So within that phospholipid bilayer of the inner membrane are these compounds called the electron transport chain, or electron transport system. What kind of molecules are they? They're proteins. Okay? There are several types of proteins. Including a group of proteins called cytochromes. This used to be referred fairly commonly to as the cytochrome system. That name's become kind of out of vogue. Cytochromes are iron-containing proteins. And that iron atom can pick up electrons and become reduced and donate them and become oxidized. Can you think of another iron-containing protein in your body that can become oxidized and reduced? Hemoglobin, right? Okay. So these behave somewhat the way that hemoglobin does. This is an illustration of the electron transport chain or system. And these each represent a different protein in that particular system. And these that are indicated CYT are the cytochrome proteins. I typically like to use this row of the membrane as representing the cytochrome system, because of the way that these chairs are tiered. Okay? So the top of the cytochrome system is up here. The last compound, the cytochrome A3, is down here. This is higher energy than this is. And so these compounds pick up electrons from reduced NAD or reduced FAD. And when this compound picks up the electrons from reduced NAD, this is oxidized again, right? That's recycled. So that job has been done. But now when this picks up the electrons, what happens to it? It's reduced, okay? In a reduced state, can it pick up more electrons from other NADHs? No. So it's got to get rid of its electrons. So it donates them here. So now this is oxidized and this is reduced. But if this is reduced, can it accept more electrons from this? No. Have you ever seen an old Western movie where maybe a building is burning down and there's a horse trough with water in it? and they form a line of people called the bucket brigade, and somebody scoops out some water and hands the pail, hands the pail, hands the pail, and then they throw it on the fire. Okay? Well, what if somebody in that particular bucket brigade doesn't throw their pail to the next person? Then you're holding a pail, you can't take another one, right? That's equivalent to being reduced in the electron transport chain. So unless electrons keep flowing through that chain from one compound to another, it's going to back up and stop if that doesn't happen. Well, it's got to end someplace, right? Where the electron transport chain finally ends up is this final compound right here, dumps its electrons to oxygen. When oxygen picks up its electrons, it becomes reduced into water. And as long as we've got oxygen present to keep grabbing the electrons from this chain, and electron donors like reduced NAD and reduced FAD donating to it, this keeps basically flowing. It keeps running. What happens if we're deprived of oxygen? Then this becomes reduced. This becomes reduced, this becomes reduced, this becomes reduced. The whole system backs up. And now these don't have any way to dump off their electrons. If that happens, if we're deprived of oxygen, what happens to this reduced NAD and reduced FAD? It's going to have to go back out in the cytoplasm again and dump off those electrons during fermentation. Because these have to be recycled. It's just a matter of how you do it. Well, as these electrons move from one compound to another, for some mysterious reason that I'll explain in just a second, we make a hydrogen ion gradient. So this is the electron transport chain, beginning, end, oxygen. As long as the electrons are flowing through this chain from compound to compound, we pump hydrogen ions out here at the expense of in here. So this space accumulates H pluses. They are depleted in this matrix. And then, if they're ATPase channel proteins in the membrane, then these H pluses can go back to lower concentration and we can make ATP. That's going to stop if we don't pump those H pluses back out here again. What does the pumping the electron transport chain does? Everything.
So the electron transport chain reduces the oxygen and it goes out into the intermembrane space.
Mm-hmm.
Okay.
And then the oxygen will diffuse out of the mitochondria into the cytoplasm, out of the cytoplasm into the circulatory system. Okay.
Okay.
This water that's produced during cellular respiration is sometimes referred to as metabolic water. So it's actually produced during metabolism. There are some desert organisms that are so well adapted to conserving water. They've got all these adaptations to keep them from losing water through sweating, for example, that they literally hardly ever have to drink because they produce enough water through metabolism to basically satisfy the needs of the body. So, we're at the point right now where we have to try to rationalize. Why is it that electrons moving through this electron transport chain makes this hydrogen ion gradient across that membrane? Okay? Why do hydrogen ions build up here and get low here? We said it's because of the electron transport chain, but why? What's it about that chain that makes that happen? Okay? Okay, look at what we're feeding into the electron transport chain from this coenzyme and this coenzyme. we're feeding in hydrogen. Okay? What is a hydrogen? It is a proton and an electron. If a compound grabs the whole hydrogen, it becomes reduced, right? If it grabs both the proton and electron. What about if it only grabs the electron? Is it still reduced? But if it only grabs the electron, what's left over? A proton. Okay. So, there are two factors that contribute to the electron transport chain producing an H plus gradient. Some of these compounds only accept and donate hydrogens. They're very picky and they'll go, I only will deal with hydrogens. Some of them will only accept or donate electrons. And these two compounds tend to kind of alternate in the chain. Second factor. These two types of carriers are asymmetrically distributed within the membrane. What's the word asymmetrical mean? What does symmetry mean? You ever heard that humans tend to think faces are beautiful if they're very symmetrical? Kind of equal on both sides. And that asymmetry, unequalness, is seen as more, less beautiful. So asymmetrical means not equal, unequal. Yeah.
These two what carriers?
These two things, these two compounds, that some of which accept hydrogen only, some of which accept electrons only. These guys right here, okay? Okay. So, if I were using the people in this row as the electron transport chain compounds, they're not all lined up just in the middle of that phospholipid bilayer, okay? Some of them tend to face this side. They're real close to this half of the bilayer, facing this intermembrane space. Some of them are like Jesse. They tend to face more the matrix. Okay? So that's what I mean by asymmetrical distribution. There's one more thing that's not part of the electron transport chain that we have to throw in here that's going to help make this make sense. What do water molecules tend to spontaneously do? They tend to dissociate, remember? And what is produced when water molecules dissociate? Protons and hydroxide ions. Now remember, this is happening. It doesn't happen very fast. The rate of dissociation of water is very slow. And that's why, if you just let water sit, you don't produce many of these. Remember what the concentration of H plus ions is when water dissociates? Remember, it's 1 times 10 to the minus 7th molar. And what's the pH of that solution? A minus log of 1 times 10 to the minus 7, or 7. Okay? So pure water slowly dissociates and produces small amounts, but they're still there, of these protons. Well, there's water in the matrix of this mitochondria. This is a fluid-filled environment. And so that water is going to be dissociated. Okay? And that's going to provide some protons. So, here's the electron transport chain right here. Here's the inner membrane of the mitochondria. This right here. Okay. Located within that inner membrane are some compounds that can accept and donate hydrogens and electrons. These are the electron transport chain compounds, okay? Here we've got NADH that is being produced by, what two processes produce NADH? Glycolysis and citric acid cycle, okay? Which one produces reduced FAD? Citric acid cycle. Okay, so those are accumulating as glucose is being oxidized. And then as acetyl-CoA is being oxidized, okay? So they're accumulating. Okay. And we need to recycle them. Okay? So, reduced NAD donates its electrons a little bit different place in the chain than reduced FAD does. Remember, this is higher energy over here, this is lower energy over here. Reduced NAD feeds in higher on the chain than reduced FAD, and that's one of the reasons why this gives us more potential ATP than that does. It gives us 3 as opposed to 2. Okay? And then as those hydrogen electrons are passed back and forth from compound to compound, we pump hydrogen ions out here. From here. And then, when we build up hydrogen ions, if there's this ATPase channel protein, they're going to flow back to their lower concentration again. And as they flow through that channel, they can make ATP. Here's another number you need to remember. For every 2 H pluses that flow through this channel protein, we can make an ATP. Okay? The electrons from the electron transport chain are eventually picked up by oxygen to make water. And as long as we've got oxygen and a source of electrons coming from the oxidation of food molecules, this keeps pumping along, pushing hydrogen ions out here from here. And those hydrogen ions can be used as a source of energy to make ATP, and then we got to pump them back out again. So you've really got all pieces of the puzzle, but next time I want to kind of add this to the mix and show you a different representation of this that really does illustrate why this electron transport chain pushes these hydrogen ions out here from in here. You've got the information you really need, but I need to probably show that to you before it's going to really make any sense. Okay? So what we will do is on Tuesday, oh wait, do we have anything? Do you have anything on Monday to do? I mean, other than the quiz? Do you have any lab intro?
I can do it at the beginning of lab.
Okay.
If you want to use Monday.
Okay, so here's what I'm going to do. On Monday, I'm really going to do a little bit of lecture along with the quiz so we can try to finish this up. That'll give you some break between the material on the exam and the exam Friday. So we'll kind of finish this up and do a little bit of accounting of how this all adds up. And if things work out the way I hope we will, then we'll be done with lecture material on Monday. We'll start some new stuff on Tuesday, photosynthesis, but that would be on the next exam. Okay. Or would you prefer some review on energetics? Okay, let's do that. Let's do that. So Monday, I'll put together some questions, review on energetics, delta G's, exergonic, etc. And then we'll do the quiz. And then Tuesday, I will wrap up with respiration. So we'll be done with the material on Tuesday. Okay? Okay? Good break today. Have a good weekend.